So, I was discussing about the osmoregulators as well as osmoconformers. Osmoregulators are those organisms which actively themselves control the salt concentration despite the salt concentration in the environment. Their body osmolarity stays constant. For example, freshwater fishes. The osmoregulator expend energy. एक्सपेंड एनर्जी का मतलब खुद अपनी एनर्जी का खर्चा करते हैं. They expend energy to control water uptake and loss in a hyperosmotic or hypoosmotic environment. Most freshwater organisms as well as marine organisms like fishes, they are stenohaline. Stenohaline. Stenohaline is made up by two words. Steno means narrow and haline means salt. So, stenohaline organisms are those organisms that cannot tolerate wide fluctuation in the salinity of the water. Freshwater organisms are stenohaline. It means that they will die in salt water. Similarly, most marine fishes or organisms are stenohaline. It means that they cannot live in fresh water. Now there are another type of animals, organism known as the osmoconformers. Osmoconformers are able to match their body osmolarity, that is salt concentration, to their environment actively or passively. Osmoconformers consist few marine animals. They are isoosmotic with their surroundings and do not regulate their osmolarity. Example, marine invertebrates. Some fishes living in fresh water tend to take up water by the environment because the environment is relatively hypotonic. In such situation, they do not drink much water. Instead, pass a lot of dilute urine to achieve electrolyte balance by active transport of salt through the gills. When they move to hypertonic marine environment, these fishes start drinking water. They excrete excess salt instead of water through their gills and their urine. Most marine invertebrate, on the other hand, are isotonic with sea water. Their body fluid concentration conforms to change in the seawater concentration. Cartilaginous fishes consumption of salt composition of blood is similar to the bony fishes. However, the blood of cartilaginous fish, for example shark, contains an organic compound urea and trimethylamine oxide. This does not mean that their electrolyte composition is similar to that of the sea water. They have achieved isotonicity with seawater by storing large concentration of urea. These animals secrete urea as nitrogenous waste are called ureo, urico, ureotelic, as these animals secrete urea as their nitrogenous waste. The marine bony fishes are hypoosmotic to seawater. They It means that the salt concentration in the cells of the fish is less than the salt concentration outside. It means their water concentration is more, hence, they can lose water by osmosis and gain salt by diffusion from the food. They balance water loss by drinking seawater and excreting salt, as I have already said before. This is a diagram where the osmoregulation in marine fish is shown. Gain of water and salt ion from the food, gain of water and salt ion from drinking seawater, excretion of salt ion from the gills, excretion of salt ion and small amount of water in scanty urine from kidney, osmotic water loss through gill and other part of the body surface. Fresh water, as I have already said, that freshwater animal, they constantly take in water by osmosis from their hypoosmotic environment. They lose salt by diffusion and maintain water balance by excreting large amount of dilute urine. The salt loss is lost by diffusion, are replaced in food and by uptake across the gills. This is a diagram which shows osmoregulation in fish living in fresh water. They gain water and some iron in the food. Uptake of salt ion by gills. Osmotic water gain through gill and other parts of the body. Excretion excretion of salt ion and large amount of water in dilute urine from the kidney.